Okay, well, hopefully you all just saw a little thing say you're being recorded. I just started now, so I don't forget. But thanks for inviting me. It's great to see so many familiar faces. Harvey, I don't think I've said hi to you yet. Great to see you. Howard, good to see you too. I don't know if you remember my little known fact, my other passion is plants. So you were gracious enough to give me some primrose for my old house before I moved. So I was delighted to see your garden a long time ago. Scott, colleague here, it's nice to see. And I think Evan, who's on here, maybe Beverly's new PhD student. So this will be great, who will hopefully be collaborating with us in this research in Bolivia. I know we've got a really diverse group. And what I've tried to do with this talk is step back a little bit and provide a little context for why we do what we do. You know, my specific area, my real passion for research is extinct mammals of South America, some of which are really obscure, even within the obscure world of extinct mammals of South America. But I hope that by the end of the day, you'll be convinced that there are some really useful reasons, not just intellectually, but also societal reasons to be interested in these sorts of things. And I'm going to pull in information from a lot of different fields, because one of the things that I've really enjoyed doing is collaborating with all sorts of experts. And as I think I mentioned earlier, Beverly Saylor has been working with us in Bolivia for a while now and has really helped introduce us to other experts. And we're pulling in all sorts of data, and that really makes, I think, our analyses a lot more interesting. So we'll have some background, and then I'll dive in a little bit more depth to our research in Bolivia, where I've been working for about 20 years now. We'll talk about some sites where we spent a lot of time, and then this main site, Nazareno, that we're targeting right now. We've been a couple times, but we hope to get back to. And with that, let's see. I'll pull up my slides.
Yeah. Look at that.
All right. There we go. So really the upshot is reconstructing ancient ecosystems. And we're focusing on the present day Bolivian Altiplano, which today looks like this: high, dry, beautiful place to do field work. But we uncover not just extinct animals, but we also try to reconstruct what their habitats look like. Oh, Peter, we might need you to mute if you could.
Cleaning lady is coming late.
First, I'll start with maybe the animal in the room. I guess I should have said I'm in the School of Medicine right now, and that's where my position is. And it surprises a lot of people when they hear that I'm in the School of Medicine as opposed to geology or biology. And the reason for this has to do with paleontology in general. If you study extinct vertebrates, animals with backbones, you have a really good idea of their anatomy, and it's essentially the same bones and muscles and everything else that we have that you'll also find in a dinosaur and a crocodile and a snake and an extinct mammal. And therefore, if you're a vertebrate paleontologist, one of the useful things that you can do is teach human anatomy. And that's what I spend a fair bit of my time doing here in the School of Medicine. I'm involved in teaching to the medical students, as well as our graduate program. We have a master's in applied anatomy, which is a really fun and successful program, mostly for students who want to go to medical school, but need to beef up their CVs, improve their MCAT scores, or things like that. So in addition to teaching for the medical students, I direct our gross anatomy course. Scott Simpson, who's on here, participates in that. It's a team talk course. I also do a couple surgical courses, which are cool. And those are ones that I organize, and then I bring in our clinical faculty from UH and other places. One of them is a survey course in the summer where students can sample 7 different specialties. We have faculty that come in, and students perform bread and butter procedures on cadavers we've got a really nice surgical simulation lab that's a shared facility down in the basement of the BRB. And then I do an advanced head and neck course. That's really what I'm excited about in terms of anatomy. Most of my teaching is head and neck anatomy. It's really complicated. There's a lot going on. It's really useful if you're interested in fossils, because you can get a lot of information both about evolutionary relationships as well as ecological adaptations from looking at skulls and teeth and things like that. And the surgical course, then, the advanced one that I organize, is all on head and neck procedures with neurosurgeons and ENTs and plastic surgeons and radiologists. And it's a lot of fun. Those then are my marketable skills. I will say I also teach every other fall a mammal diversity and evolution course. And Evan's taken that. It's mostly for biology undergraduates, or at least those are mostly the students that take it, and we'll get about a dozen or 14. And then typically we'll have a handful of graduate students that will also take it, students in biology, geology, which is earth, environmental, and planetary sciences, and then ones that are working with me in the anatomy department as well. But it takes a lot of time, so I do that just every other fall. Mostly I'm teaching human anatomy. And paleontology is cool, I think, because it's really at the intersection of these two huge fields, right? On the one side, we have biology, and on the other side, we have geology and the geological sciences. And these are really the two starting points where you can head towards paleontology. Most paleontologists start as one or the other and then head towards the middle. I was trained as a biologist. I got my PhD at the University of Chicago, and that's a place where the paleontologists teach human anatomy. That's where I learned human anatomy and started teaching it. But, you know, depending on where you are, you may start in a geology department and work your way towards paleontology too. And therefore, no matter which side you start on, I find you're always trying to learn more about the other one. So in my case, I'm always trying to learn more about the geological sides of things. And we can pull all this together into what I like to call paleoecology, which is really the idea of not just looking at the animals themselves or the specific group you're interested in, but really trying to paint a picture of what a past ecosystem looked like. And it takes both sides of this diagram to be able to do that. So reconstructing ancient habitats and ecosystems is what we're going to be focusing on today. And I'm not going to get too far into the details on the particular mammal groups or taxonomy or anything like that. Although, of course, I'm happy to talk more about that in detail. Although, of course, I'll also show some fossils and reconstructions and things like that. The idea with what we do then is that we'd like to be able to go to a place like this. This happens to be a site called Tingarica in central Chile, near the town of Termas del Flaco. That's what you see in the front there. And those dark rocks in the middle, especially the ones in the far back there near the horizon, that's where we find fossils that are about 31 million years old. They correspond to the early Oligocene age. Ideally, what we'd like to do is go to a site like this, collect fossils, collect geological data, and be able to paint a picture like this that hopefully is accurate as it can be in terms of the vegetation that's there, the way that these fossils accumulated, the animals that were there, some idea of the temperature, moisture levels, and things like that. To do that, we have to collect lots and lots of different types of data. But you might also ask, well, why do we want to do that? It's nice to have a picture like this. I think you'd agree. But of what utility is this in the greater scheme of things, other than for talks like this and for training students about life in the past? Well, hopefully global climate change is not something new to this audience. We're all familiar with it. And, now it's more and more that we hear about it on the news with extreme weather and things like this. And so if we compare temperatures in the last 10 years to historical data, we can see that overall, most areas of the globe are experiencing increases in temperature. And, we use data from the past to try to reconstruct temperatures in the future. And what I'm going to, hold on one sec. I need to hide my screen controls here because they're covering up my titles. Let's see. There we go. In climate models, we hear a lot about in the news, and we use these to predict what's going to happen in the future. And in the news, we hear, if we can restrict increases in temperature to a certain amount, this is what we can predict. And that's all based on models. And we can model temperatures in the future. You can see here ones that are modeling it for near 2080 to 2100, as well as precipitation. Testing these models relies on paleontological data. And of course, your models are only as good as the data on which they're based. And then once you create those, hopefully you can take other data, test them, and get reasonable results. And that's really where paleontological data are useful and indispensable. This is a graph of temperatures, and you can see where we are now and then some potential temperatures in the future, depending on certain scenarios. And those are far above the average of what we've seen since humans have been on this earth. And if we want to be able to model temperatures that are really getting that high, if you look towards the left side of that graph, you can see that we can't just look at tens or hundreds or thousands or even hundreds of thousands of years ago. We really need to get into deep time, get into millions and tens of millions of years ago, which is where we work. But I also want to point out that although temperature is important and what we often focus on, there are many other variables in climate models that are equally important, if not more important. Of course, they're all related. One of these is carbon dioxide, something else that we hear a lot about. This is a major greenhouse gas. And it's interesting because its atmospheric concentration has varied substantially over the past 66 million years, which is the time since the dinosaurs went extinct. And we have a little graph here on the bottom. On the left is old, so that's 66 million years ago. And then as we come towards the right, you can see where we are now at human settlement. Relatively speaking, we are at lower carbon dioxide levels. Now, of course, with fossil fuel consumption, that has started to change, and the possible futures have it going up really, really high. How is this going to affect climate? Well, if we want to look at examples in the past, we have to go far back in the fossil record to get those sort of data and to try to correlate higher levels of carbon dioxide with other characteristics of climate. Ocean currents are also important, right? They absorb and distribute heat as well as cold, and a lot more than air does, right? Or it's a different order of magnitude in terms of how much they can absorb at the rate they do it, but they're super important. You know, the Gulf Stream that brings warm water up north, the cold water that brings cools the California coast right near San Francisco and farther down. These are also very important, and how these work depend on winds as well as the positions of the continents through time. This is a picture of not the continents today, but how we think they were about 50 million years ago. And in some ways, it's hopefully very familiar. You can still recognize the major continental land masses, but in other ways, it's quite different. You'll see some places submerged, some places are connected that weren't before. I'll point out one area right here, which is interesting. This is the southern tip of South America, which was connected with Antarctica until probably around 35 million years ago. And to illustrate the effects that currents can have on climate, what we think happened is once this connection was finally severed, that started to permit currents going around Antarctica, the circumpolar Antarctic current, which isolated it thermally. And that's really what started the process of permanent glaciers on Antarctica, which weren't there beforehand. That had a global cascade and really was a major shift, tipping the Earth from a greenhouse where relatively warm temperatures all over the place, forests all the way down to southern South America, and even there in Antarctica, to more of an ice house, more similar to what we have today, where you get much more dramatic temperature differences between the equatorial regions and towards the poles. And that's just one spot, one relationship between these continental land masses. Of course, it's tectonic plates that are responsible for the moving continents. Hopefully that's a term that's familiar to all of you. We have a series of large continental plates and oceanic plates, as well as many smaller ones in between that have complex movements, and that's how these continents can shift. But importantly, these also create mountains. Here we're looking in a two-dimensional map, but if we do a cross-section, we can start to look at topography and how those are affected by moving continents. One of the classic ways that mountains are formed is through subduction. And that's where one plate, typically an oceanic plate, gets pushed down beneath a continental plate. That's going to accrete material, which will contribute to mountain building, but also it's going to create volcanism. And these are a couple examples of volcanoes from South America. South America is the classic example of mountain formation through subduction, and that's what has formed the Andes Mountains. The Andes Mountains are the longest mountain chain in the world. They run for nearly 9,000 kilometers all the way down the west coast of South America, curving from Venezuela in the north all the way down to Tierra del Fuego there in the south. Because these are of subduction mountain chain, there are lots of volcanoes. There's a northern volcanic zone, there's a central volcanic zone, there's a southern volcanic zone. And I highlight these to point out the fact that although we talk about this as one process, it's really pretty complicated because not everything is moving at the same rate, at the same time. It turns out the angles of subduction can be different. And it's not as if the Andes rose to their present height all at once. So we have to focus in a little bit on these different areas. Some areas we have less volcanism, some we have more. In that area of Chile that I showed earlier, where we had the reconstruction of that 30-million-year-old site, that's in the southern volcanic zone. One reason why we had a volcano shown there in the past. Those volcanoes have been erupting for tens of millions of years. In Bolivia, we're up near that central volcanic zone, closer to the southern part of the tropics. We talked about ocean circulation patterns being important for climate, but air circulation patterns are also very important. We have the Hadley cell, which has been in the news earlier this summer, which goes from the equator up to the 30 degrees north. We have other cells in our region, and then these polar cells up near the north. These are also important for circulating. These are created by the winds and the rotation of the globe. And these air patterns not only distribute temperature, but they also dictate precipitation, right? And our weather typically comes from the west here, although we can get examples where it's going to be coming up from the southeast. And this all has to do with these main directions of winds as well as these air cells. However, this is a simplified view that we're looking at here. This is what you learn if you take Geography 101 of how these things work. These are affected by topography, especially mountains. And mountains can create what's known as rain shadows. That may be a familiar term to you. Essentially, when air rises, it cools and it holds less moisture. The moisture is going to fall as it rises up the side of a mountain. And then on the opposite side, the air that comes down is going to be very, very dry, and we tend to get deserts on the leeward side of the mountain. So they can have lots of effects on winds and on precipitation. Therefore, if we want to be able to create these models in the past and use them to extrapolate to predict climate in the future, a key question is, when do the Andes Mountains form? Because that tells us when we need to start taking their topography into account in these air circulation patterns, in these precipitation patterns, when modeling things like temperature and carbon dioxide in the past. And that's where we, as geologists and paleontologists, come into the picture. But we can't just go back in time and measure the Andes Mountains. How do you measure elevation in the past? Measuring elevation now is pretty straightforward, particularly with a lot of the electronics that we have. But measuring in the past is really a challenge. Even though it's simple in principle, it is challenging in practice. Fundamentally, what we look at is temperature. And this has to do with what's known as the environmental lapse rate, which means, what it is the phenomenon that for every 300 meters you rise in elevation, the temperature decreases by 2 degrees Celsius. So for 1,000 feet, it's about 3.5 degrees Fahrenheit. If we look at a mountain then here that is close to 20,000 feet, this is how temperature would change. If it's a comfortable 81 degrees Fahrenheit, 27 at sea level, by the time you climb to the top of that mountain, it's going to be almost 10 degrees Fahrenheit. It's going to be well below 0 Celsius. This hopefully isn't surprising. This is why we get snow-capped mountains, right, that are there all year round. But most time people don't really stop to think about the fact that this is a very systematic relationship. And there are lots of other associated changes with this. But the idea is that if you can measure temperature in the past, you can get some idea of how high you were. Well, how do we measure temperature in the past? And there are several different approaches to this. One that's been used a fair bit is fossil leaves. And this is an example of some leaf macrofossils. And hopefully in the upper left there, you can recognize this as a leaf. That's a fairly complete one. We also have fragments here. And there are folks that specialize in fossil leaf, paleobotanists, and can use those to try to get at temperature. And that's because the edges of the leaf, what's known as the leaf margin, there's a relationship between that, or at least the proportion of leaves with a certain sort of margin, and temperature. On the left there in A, we have what's called a complete margin. It's basically even, whereas in B, it looks like a little saw blade, and that is a serrated margin. In habitats like ours, temperate zones in Ohio, you're going to see many more plants that look like B than look like A. Think about oaks, think about maples, think about beeches, etc. The opposite is true in the tropics, where you get warm temperatures and lots of precipitation. Leaves tend to have smooth margins. And here we have things like magnolia trees, which have a smooth margin like A, but they're relatively not very diverse, whereas most trees are going to look like that in the tropics. We can go back to fossils like C and D. We can look at the proportions of leaves that have these different sort of margins and get some idea of temperature in the past. And this phenomenon with vegetation changing with temperature can also be extrapolated to elevation, right? This is a beautiful drawing by Alexander von Humboldt from 1850, who is one of the first people to really recognize the fact that as you go up in a mountain, it's also sort of like going from, well, he was in the tropics. This was done in Ecuador, right, from the equator, which is right at the tropics. But going up a mountain in the Ecuador that looks like this one, well, he has several ones drawn on here or illustrated. Chimborazo is a volcano that's right near Quito. This is a place where you can get up to 21,000 feet. So you can imagine this being Chimborazo, and you can hike up it, and it is like hiking from Ecuador all the way down to Tierra del Fuego in terms of the changes in vegetation. And that is related to this change with temperature as you go higher. We can also use stable isotopes, such as oxygen. For those of you that aren't familiar with isotopes, isotopes are elements that have different numbers of neutrons, and so they vary in their molecular weight. Isotopes of oxygen can be oxygen 16, like we see here in yellow, or it can have a couple more neutrons and be oxygen 18. And different isotopes of the same element will have slightly different properties. They behave differently due to that different weight. And oxygen can be a very useful one because it is involved in precipitation. It's the oxygen that's in water, which is, of course, H2O. And without getting into the details of this, you can look at the proportions of these two different isotopes of oxygen, oxygen 16 and oxygen 18, to give you an idea of temperature, because they behave differently in terms of how they evaporate, how they rain out. They don't evaporate and rain out in equal proportions. They're in different proportions. And that also varies with temperature. It really gets pretty complicated because there are lots of other things that affect it too. Suffice it to say that there are people who are experts in stable isotopes, and these are called stable because they will maintain that molecular weight for tens of millions of years, so you can measure them in the past. People that are experts in this can pull these sorts of data out from samples that we take from various paleontological and geological sites. This is just a diagram of the proportion of this heavier oxygen isotope on the Earth today. And what I'd call your attention to is that not only is this related to temperature, right? It gets bluer near the poles. Note that we have lower delta 18 values also in the Andes. It also changes with elevation. Therefore, you can use oxygen isotopes to try to get at elevation as well as temperature, via temperature, of course. And as I mentioned, since these are stable isotopes, we can collect these data from fossil teeth and bones, because animals that drink this water incorporate those oxygen isotopes into their teeth and bones. Typically, teeth will work better for preserving this record because they're essentially rocks and they don't change geochemically over tens of millions of years. Bones usually are a little bit more porous and they can change. But looking at both can help you distinguish conditions when those animals were living from those in the environment in which they were living that might have changed subsequently. With that background, let's take a look at what people have come up with for elevation in the central Andes thus far. And this is mainly based on fossil leaves and on these stable isotopes. Now, I know this is sort of a complicated diagram. This is not the way I would have done the diagram, but I just pulled it from a really nice review article. The three columns are the northern, central, and southern Andes, which I talked about before. You can ignore the first two because we're going to focus on the southern Andes. And this is a little bit backwards from how we think of things in geology, but oldest is at the top and youngest is at the bottom. And the modern elevation is there in blue. So just where this arrow is, pay attention to those two dots right there. And those show two different ways to elevate, or to estimate what we think the elevation of the Andes was in this 24 to 16 million year gap, which is a pretty big gap. But if you look at the key on the far left there, you'll see that this area is today at about 4,000 meters, if you look up to that blue line, and back in the early Miocene, which is what this corresponds to, it was less than half of that, probably between perhaps 1,000 and 2,000 meters, a lot lower. The problem is it's more complicated than we thought, which is maybe always the way in science. It turns out that surface temperatures vary with the height of the Andes. And these are models showing temperature based on different assumptions about how high the Andes were. And what this means is that the starting point for calculating elevation depends on elevation, which is a little bit of a catch-22, right? Like, how do you figure that out? In addition, winds, as well as the sources of moisture, can vary with the elevation of the Andes. This is important because this affects the starting point for some of these isotopic analyses. When we think about oxygen isotopes, you have to start with what you think you know about where that water started. And if it's coming from the Pacific, that's different than if it comes from the Atlantic. And that depends on the Andes, which affect the winds. Well, how can we improve predictions given all of these complicating factors? The approach that we've been using and that we hope to continue applying is really a multi-proxy approach. We're using lots and lots of different types of data to see if they can give you a concordant answer, not just relying on the plants, not just relying on isotopes. And fossil mammal sites are great because they allow us to collect many different types of data. Bolivia has many sites that are suitable for such analyses and is one reason why we spend a lot of time working there. And here's a little map of Bolivia. We're about 20 degrees south, puts us just in the southern part of the tropical latitudes, with Bolivia there in pink. It's one of two landlocked countries in South America. And in orange here, I've pointed out 3 sites that we have worked at previously, as well as in blue, another site very close by in northern Chile, which used to be Bolivia way back before the War of the Pacific, and now is in northern Chile. And that's another fossil mammal site where we have worked. Of those, I want to start by just giving you a little bit of a review about Quebrada Honda, which is where we have worked the most out of those 4 sites. And I think we have the best idea on the animals that were there and the sort of habitats that they were living in. And this is a picture of the outcrops at Quebrada Honda. It's another really beautiful site to work at. If you look closely, you can see a couple paleontologists, geologists there in the foreground, either excavating something or taking samples. And Quebrada Honda, we zoom back out. Here's where it is in South America. It's in the southern part of Bolivia, very close to the border with Argentina. The Andes, we talk about the Andes mountains, but in this area, they're really very wide. You know, it's not just one mountain chain. It can have several sub-chains to it. And this is in the eastern Cordillera of the Andes Mountains, which isn't the highest part. It's the slightly lower part on the eastern side. And just to the west of that, this big flat area is the Altiplano. And that Altiplano is about 4,000 meters today. And the eastern Cordillera of the Andes, where this site is just a little bit lower, about 3,600 meters or so. We have some good age data on this, particularly thanks to a couple studies that we've published in the past couple of years. And the site ranges from about 13 to 12 million years ago, which puts it in the late middle Miocene, if you're familiar with the geologic timescale. My history working in Bolivia goes all the way back to, 1999. This was when I was still a graduate student at the University of Chicago. And I was invited to a conference in La Paz, Bolivia, which was the first and I think only conference, Evolución Neotropical del Cenozoico. So basically, neotropical evolution of the Cenozoic in Bolivia, that was put on by a guy, Bruschaki, who was doing a Fulbright in La Paz at the time and working with the folks at the Natural History Museum in La Paz. And it was a great excuse for me to go to Bolivia, where I hadn't been before. Most of my work had been centered in Chile prior to that. But this was my introduction to Bolivia. I was able to present some of my thesis work. And the most important part is meeting other researchers who work in the Cenozoic of South America, and the Cenozoic being the last 66 million years. And that's where I met this gentleman, my friend and colleague, Federico Anaya, who is from Bolivia. At that time, he was working at the Natural History Museum in La Paz. He subsequently moved to a university in Potosi, and that's where he is now. And we have continued to collaborate ever since that time. When he was curator at the museum, he showed me around the collections, and this is one of the specimens that he showed me. This is a jaw. The scale there is in centimeters, so it's a few inches long. It's a lower jaw of a plant-eating mammal. It belongs to a group of mammals that's totally extinct. It was a very diverse group of mammals known as notoungulates, plant eaters that lived in South America. They originated there, lived there for tens of millions of years, and then they went extinct. So we don't have any more notoungulates. They were only found in South America, with one small exception. And this belongs to a particular group that I was really familiar with, because my thesis focused not exactly on this group, but they're very close relatives in older rocks. But I recognized that this was something new. This was not any species known so far. And I was obviously very enthusiastic as a graduate student. Oh, there's a new species. And there's a beautiful specimen, virtually complete. And there were other specimens, parts of the top part of the skull, the cranium. And I told Federico, we've got to write this thing up and publish it. And we eventually did that. It takes a while. Got to finish the dissertation. You have other obligations. But it finally came out in 2006. We published it in the Journal of River Paleontology, and we named it Hemihegeditherium trilobus. And trilobus, that was the new species. The genus, the first part wasn't new, but trilobus referred to the last tooth, which had 3 lobes. I'll just back up here, and it's this tooth right here. You can see it has 1, 2, 3 parts to it. All other close relatives just have 2. This back part is all one big piece right there. And that's why we called it Hemihegeditherium trilobus. And this is a nice reconstruction by Velizar Simeonovsky. You're going to see that name a lot. He is a Bulgarian zoologist who is a research associate at the Field Museum in Chicago. We got to know each other when I was in Chicago. And he's an artist. He's a fantastic artist in addition to being a zoologist. And we've collaborated on doing reconstructions of lots of these extinct South American mammals, because I think for a lot of people, it's tough to visualize what a mammal looks like from just a jaw. And maybe one should never do that. But this gives you hopefully an idea of what we think this animal might have looked like, a dog, small to medium-sized dog-sized ungulate, little hoofed mammal, something in between a rodent and a small ungulate. We then wanted to go to the field, and we first went to the field in 2007. This was the site of Quebrada Honda, and interestingly enough, I went with Carmi Garzioni here, and if you were, look at all the slides I show very carefully, you might have noticed Garzioni was the name on that graph that I showed of the elevations of different parts of the Andes at different times. She was interested in taking samples for some of her studies on elevation. She is an expert in isotopes and uses a certain type of isotope called clumped isotopes to try to get elevation. We went together and went to this fossil site. Of course, I was interested in the fossil mammals. We found Hemihegiotherium trilobus specimens like this. That's Federico that's holding it. We found lots of other great fossils there too. Carnivorous marsupials. South America was an island continent for most of the time between when the dinosaurs went extinct and when the Panamanian land bridge formed about 3 million years ago. Therefore, there weren't any cats, dogs, bears, weasels, raccoons, anything like that on South America. And that niche of meat-eating mammal was filled by marsupials, just like it is in Australia today. And this is one specimen that we found. It turns out they're very, very rare. About 1 in every 100 specimens is a carnivorous marsupial, if you're lucky. This one, we're looking at a couple leg bones here. These are some vertebrae. We also found part of the jaws. And this is one that's called A. scion mectoteros. I like to describe it as a weasel dog. In its skull is dog-like in having kind of a long snout with spaces between the premolars, but it was not a long-legged animal like a dog. It had much shorter legs, something more like a weasel. We don't really have anything like it today at all, so reconstructing it is a challenge. And it's fun working with Belazar to think about what these things might have looked like. Armored mammals are things that we find at Quebrada Honda. These include armadillos, which are familiar to all of you, I'm sure, but also things like this, which are glyptodonts. And I love this picture because it allows me to tell what I think is kind of a funny story. This was not a photo that I took. This was taken by one of the graduate students of Carmi Garzioni back there in 2007. This is his rock hammer. And at the end of the field season, when we're back in La Paz, we're trading photos, and he shows me this photo. He's like, oh yeah, look at this really interesting rock that I found. I took a picture of it, and I said, that's not a rock, that's a glyptodont. What we're looking at here is the shell of what was essentially the mammal equivalent of a tortoise. These were animals that were larger to much larger than armadillos. The biggest ones got to the size of a small car, so they weighed a ton or two, and their shell was totally fused up like a tortoise's. They couldn't roll into a ball. And what we're looking at here is just part of that shell. All those little bones there are separate bones that get fused together, and they're starting to erode out. Fortunately, Dave had taken the GPS, and we were able to go back several years later and take this specimen out. With this specimen and some other ones that we found, we haven't written these up yet. It's a new species, collaborating with some folks in Argentina to describe it. But one of the really neat things about it is its head, because it is characterized by having 4 horns on top of it. This is the first horned glyptodont that will be described. It turns out that there are horned armadillos that once lived in South America, which are really cool. Now, these aren't horns like the long horns that you're going to hear about next month, or even the antlers like we have on deers. These are really just bones of the shell that are modified into horns on the head, but they probably functioned like horns. And we have in these glyptodonts a single bone that has these 4 points that would have sat right on top of the head. And we know that because we found it with skull remains right underneath it. At some point, we're going to describe it, but this is Belazar's reconstruction of what that thing might have looked like. Rodents have been in South America for probably about 30 million years or so. And today they're really diverse. You get cool ones like chinchillas and capybaras. This is an example of a tiny rodent jaw that we might have found. This was a new species that we described from Quebrada Honda. Quebrada hondomes is what we called it, the mouse from Quebrada Honda. Although it's not a mouse, it's more closely related to a group of animals known as spiny rats that live in tropical areas of Central and South America today, many of which are really pretty attractive as far as rodents go. The striping is not something that we just pulled out of nowhere. You'll see this in some modern spiny rats. We also have big plant eaters. These are just a couple teeth that I'm holding up right there. These are cow to rhino-sized animals, different side of the notoungulate family tree from that Hegetotherium trilobus that I showed you earlier, but these are also pretty common. And then a different type of plant eater, this is a whole separate group, they're known as litopterns. And these were a little bit more like modern camels or deer in terms of their skulls and in terms of their limbs. The reconstruction there you can see looks very camel-like or deer-like. They're really peculiar, though, because their nostrils weren't at the tip of their snout. They were partway back, and in more recent ones, they were probably all the way back near the eyes. That's a whole talk in and of itself. They're very peculiar. But the fossil in the lower right there, what we're looking at are some articulated leg bones. It's part of the lower leg there, as well as part of the upper leg. And as an aside, I'll often have undergraduates or graduate students who work to describe these fossils. And the litoptern group, I had a really great undergraduate who worked with me on litopterns, Andy McGrath, and we ended up describing several new species of the two different subgroups of litopterns, and then he went on to do a PhD elsewhere. In total, we've described 38 species of mammals from Quebrada Honda. We have different types of marsupials. We have armored mammals, sloths, which I didn't show, but there's a diversity of sloths that have been in South America for a long, long time. And we had 5 different species there, lots of rodents. And then in the right column are all these extinct hoofed animals that were pretty common, just as plant eaters are common today. You'll see that a lot of these have species names, and many of these are new. That's one of the things that's really notable about Quebrada Honda. And that's simply due to the fact that people haven't worked there before. And when you go to new sites or you sample new time intervals in the fossil record, you're more likely to be able to find and describe new species. And that's what we've done at a lot of our sites in Bolivia. Some of these are still in process. Like the Astrapathere, we have pretty fragmentary remains, and it's tough to really get a good idea of what it is. But I have a student who's working on that right now, as well as the sloth. The Mylodont looks to be a new species, but you just have a new teeth. So it's not finished yet. A lot of the armored mammals we're also still working on with colleagues from Argentina. It's a good news, bad news scenario. The good news is we've got some beautiful specimens of armadillos, complete skulls, postcranial bones, complete shells, which is great. The downside is there's so much to describe. It takes a long, long time to publish these things as opposed to when you just have a jaw or part of the shell. How high were these mammals living then? As I said, today the site's now at about 3,600 meters, so 12,000 feet above sea level. This is what it looks like, high and pretty dry. Certainly these animals were not living at that elevation, but how much lower is really uncertain, although we think we're getting closer to the answer. This is a graph of this area of the Andes elevation through time. On the left is 30 million years ago, on the right is now, and then of course the y-axis is paleo elevation. And you'll see the different colors there are estimates based on different proxies. Blue are the oxygen isotopes, green are the fossil plants, and then orange is what Carmi Garzioni specializes in. It's these clumped isotopes, which are supposed to be better and less affected by some of the factors that can complicate oxygen isotopes. And this is her graph from looking at the northern part of Bolivia. And that's why it says clumped isotopes N. This is the northern part of Bolivia. And what you'll see basically is it's low. And then we have a period from about 25 to 10 million years where it's maybe 1,000 meters up to 2,000 meters. And then we get this big jump about 10 million years ago. And that's when it gets close to the modern elevation. That seems to be the general consensus with a caveat that the error bars on these are pretty big, right? About plus or minus 500 meters on that average. But that's what we think is going on based on the record, at least in northern Bolivia. 6 years later, Carmi published an interesting study looking at the southern sites, which included Quebrada Honda, which we just talked about, as well as Cerdas, which is another site that we didn't talk about. And she proposed a somewhat different scenario. This is the old site. There's Quebrada Honda right there, and then there's Quechua. And the pattern that she came up with was that it was asynchronous, this late uplift, that it started a bit earlier. And since this site here is Quebrada Honda, that has implications for all those mammals that I just showed you. That rather than living relatively low, 1,000 to 2,000 meters, she proposed that maybe they're up to 2,500 to 3,500 meters, not that much lower than where they are today. This also has implications for the tectonics. All of a sudden, geologists who deal with this sort of geology have to be able to explain, okay, how do we get the Andes rising earlier in the southern part of Bolivia than they are in the northern part? So this has important implications, not just for models, but all models for climate, but also model for geophysics and structural geology. In 2013, in one of our trips to Quebrada Honda, we came across this fossil, which was interesting because I didn't immediately recognize what bone it was. I mentioned that we all basically have the same bones, whether we're a dinosaur, a turtle, or an ancient Homm. But this one, I wasn't quite sure what it was. It didn't look like one of our forearm bones, like an ulna. It didn't quite look like a pelvis. I'm like, it's weird. And it's pretty big, right? You can see my finger there. And I thought, well, this is certainly worth collecting. We'll have to see what it is. But, it's not super high priority. We'll come back and collect it in a couple of days. And that we did. And once we got it cleaned off and also looked at some of the other remains that were nearby, it became abundantly clear why I didn't immediately recognize it. It's because it wasn't from a mammal. There's an outline of it just to show you this bone, what it looked like in the field. We also found chunks like this, which I also hadn't paid too much attention to. I thought, wow, maybe it's a weird glyptodont, but this is the shell of something else, and it is a tortoise. And that bone that I showed you before, the reason I didn't recognize it is because part of the pectoral girdle of a tortoise. And those are very different than what you find in mammals, because turtles and tortoises are super bizarre. Their shell is fused to their ribs, and their arms are actually inside their rib cage. So their scapula and coracoid and all this doesn't look anything like what we have as mammals. And the size of that indicated that we weren't just talking about a box turtle or a painted turtle. This corresponded to a giant tortoise, something on the size of what we might find in the Galapagos Islands, which is what this is a picture of right there, a Galapagos tortoise with an egret there on its back for scale. In addition, that year, we found a very small turtle fossil. This is just a little chunk of a shell. You can see the 1 centimeter scale there. So this isn't bigger than 1.5 inches, but this little chunk had some diagnostic features on it. And this isn't something I recognize. This is a great example of why we collaborate. I enlisted a South American turtle and tortoise expert to help us describe these things. And he recognized it as being very closely related to some living South American turtles, specifically those that belong to the genus Acanthocheles. And these are side-necked turtles, ones that you find in the tropics of South America, not the ones that you find up here in temperate North America. This allows us to measure elevation with turtleometers, which can be pretty useful. And the way we do this is we look at where these species are found today, or at least close relatives of these species. We can look at the temperature and look at the elevation, and these graphs show for different species of tortoises in the top, giant tortoises, and these side-necked turtles in the bottom. It shows mean and standard deviation and range of their modern elevation. You can hopefully see from these that none of these today live at elevations higher than 1,000 meters. And this would lead us to suggest that probably Quebrada Honda was not nearly so high as some of these new data were suggesting, that it was probably 1,000 meters, maybe lower, maybe a bit higher than that, but certainly not close to its modern elevation. Therefore, we would alter this a little bit with our turtleometer and say that, in fact, 13 to 12 million years ago, it was lower, and that the southern Bolivian Andes probably had a pattern very close to the northern part. We do have some other animal data that suggests this interpretation is correct. This is a vertebra from a snake, which we also found that same year. We have more of this. This is part of the skeleton and something else that's still being described. And this corresponds to a big boa constrictor, something that's probably on the order of 4 meters long. Another example of an animal that you don't expect to find living high elevation in the Andes. We don't find them at high elevations in the Andes. These are restricted to pretty warm tropical habitats now. But that's not all we do. We don't just rely on vertebrates. And the whole point of this talk is to illustrate some of the other techniques that we can use to try to get at paleoenvironment. Because if we have isotopes on one hand and we have vertebrates on the other, it's a he said, she said. But what we'd like to do is really pull in other sorts of data to see if they support the clumped isotope model of relatively high elevation, or the ectothermic vertebrates, the cold-blooded vertebrates model of relatively low. And this is a ex-PhD student of mine who's graduated and now is out there teaching, Angeline Catena. And she focused her thesis on looking at Quebrada Honda and Cerdas from a couple different ways of paleoenvironmental interpretation. This is a picture of her trenching at Quebrada Honda. One thing she looked at were ichnofossils. Ichnofossils are tracks and traces of past life. It can be vertebrates, but oftentimes it's not. Here are some examples from Quebrada Honda. These are dung beetle brood balls. Dung beetles are very charismatic beetles that collect usually large herbivore dung, and then they put their egg in it, and that's what their larvae will feed on. And these look just like dung beetle balls that you might find on the Serengeti of Africa today. These are examples of bee and wasp nests. Bees and wasps, we think of bees and wasps as being social, but in fact, most of our native bees, and I don't know so much about wasps, but certainly the bees are not social. They're solitary. You may have different females depositing eggs in the same area, but they make these solitary cells, and that's what we're looking at here. These can give you an idea of where the water table is, because you can imagine that bees don't want to have their little nests submerged for very long by water. We can look at fossilized roots, which is what we see above, as well as burrows. Fossilized roots are another way to get at vegetation. Do you have lots of big trees, or is it all just small grasses and herbs? Burrows can tell you sometimes about what types of animals were living there. This one is probably a little vertebrate burrow, which probably doesn't tell us too much about environment. But if you're looking at things like crayfish or cicadas, those are a little bit more linked to things like moisture level, where the water table is, etc. Angeline also looked at the paleosols, which are ancient soils. And this is a relatively new field compared to certainly paleontology and many aspects of the geological sciences that really, only in the past 50 years has come into its own, and especially in recent decades has really become much, much more sophisticated. The short version of the story is that soils, just like anything else, can fossilize. And because soils are products of the original material, the environment they're living in, as well as all the organisms that were there, the plants, the invertebrates, the vertebrates, they're thought of now as paleocritical zones. Like, this is where all life has taken place, in this upper few meters of the soil and the area above it. And you can go through and recognize lots of features in the past. And by describing the soils and their contents, you can get a picture of the conditions that they formed within. And it can be really nice because these soils form on different time scales than the rocks layers themselves. And by pulling different sorts of data together, you can get instantaneous information with time averaging over tens of years to hundreds of years to thousands of years. And it's lots of different perspectives that you can try to pull together. But this is an example of stacked paleosols, and the ichnofossils that are there. And she put these together. She did a variety of these for her thesis. Something a little bit different that she did was what's known as an ecological diversity analysis. Now, this is one that focuses more on the animal fossils, the vertebrate fossils that we found there. And I think conceptually, this is hopefully intuitive. This is one that analysis that correlates proportions of species in different ecological categories with the habitats in which they live, which is a little bit of a mouthful, but I think an example is better. This is a monkey. Hopefully everyone recognizes that as a monkey. And if I were to ask you what sort of habitat would you expect to find a monkey, the answer I would hope for would be tropical forests, which is true. You don't find monkeys here in Northeast Ohio outside of zoos and places like that. And that's because the resources they rely on, especially fruits, have to be available all year round. Therefore, at fossil sites where you find monkey fossils, you can be pretty sure, there are always exceptions, that you're going to have trees and then it's not going to be too cold. We can do the same thing for animals like this. This is a mara. This is not an ungulate. This is basically a souped up guinea pig. It's a very close relative of guinea pig, but it's got these long legs. And those long legs are what you typically find in animals that live in more open habitats. These are what's known as cursorial specializations. If you find animals like this, maybe many different species, or maybe they're very abundant, you might assume that there must be some open habitat in the area because you don't typically see animals like this living in a tropical rainforest. An ecological diversity analysis pulls this sort of information, locomotor habit, diet, body size, from all the animals in a particular site to try to get an overall picture of what sort of habitat they were living in. Because any particular animal can lead you astray, but the idea is if you put them all together, you're more likely to get a correct answer. And we use patterns in modern faunas to apply to fossil ones in order to get at the habitat. Was it open? Was it closed? Did it have lots of moisture, or was it relatively dry? In Angeline's study then, she compared several sites in South America, you can see that are there in yellow in South America, with ecoregions from throughout the world. And that's what all those colors are everywhere else. Ecoregions in North America, in South America, etc. She took the faunal lists from those, coded all the different mammal characteristics, and then we did some different multivariate analyses to compare these fossil sites to the modern ones. We did one fossil site there in Europe, you might see, just as a test, because South America, since it was an island, there's some peculiar things about it. That again is another story, but that's why you see that one there in Europe. She looked at 179 different ecoregions from around the world. It was a fair bit of work. And then these 4 fossil sites in South America, including Quebrada Honda. And what did she find out? These are the 4 sites. Most of these we're not going to look at too closely, but I call your attention. Here's Quebrada Honda. She reconstructed it as a flooded grassland or savanna with a rainfall of about 90 to 100 centimeters per year, which is not that different than what we get here in Cleveland. This is a sort of interpretation that would be compatible with probably relatively low elevation. Phytoliths. This is another thing that we can look at. Phytoliths are little silica bodies that form in the cell walls of plants. And that's what we're looking at there in the lower right. Those are different sorts of phytoliths. These are structural silica bodies that help them, grow and maintain the form of their stems and leaves. These can be identified to group if you're an expert in phytoliths, and sometimes you can get them all the way down to species. But for reconstructing environments, it isn't always necessary to go to the species if you know what type of general plant it's from. This is something that we tried in Quebrada Honda. We took a variety of sediment samples from 4 different areas. Didn't have great luck, but we got some information. About 20 samples had enough phytoliths for quantitative or qualitative analyses. And this is a multivariate space of different proportions of phytoliths. If we simplify it down, we essentially have 2 axes here, or really 2 groupings of phytoliths. On the one side of the graph in the lower left, we have phytolith groups that are dominated by closed habitat grasses, which are bamboos. Bamboos are grasses in the taxonomic sense. These are ones that you'd more typically find in the understory of forests. The other configurations of phytoliths were dominated by open habitat grasses as well as palms. And we were able to look at the distributions of these throughout the stratigraphic column in the few sites where we're able to collect phytoliths. And this is a work in progress. So this is something that we've got a draft of a manuscript. We still need to finalize it and put all the data together to get the bottom line on what it's telling us. But on a first pass, what we could say is that it's a mosaic landscape with shifts between relatively closed habitats with bamboo groves and more open ones with grassy patches. And if you look at that diagram on the right, hopefully you can see it's not like it goes all from green to all from blue. We get variations between the two, not only in a single column, but also between two columns. And that these different columns represent different collecting areas within Quebrada Honda, which means that maybe you have a closed area, but if you go several kilometers away at that time, it might be more open. And this in general matches the other type of data that we've talked about, which indicates high seasonality. And for here, it's probably mostly moisture seasonality, wet season, dry season, probably about 100 centimeters of precipitation a year, and then a mixed to open habitat. What elevation then were Quebrada Honda mammals living? Well, we're still working on the answer, but it wasn't dry and it wasn't cold. We can say that much, and therefore we think it was probably relatively low. If it were as high as 3,000 meters, we would not expect to get the patterns that we're seeing from these different sorts of information. And this leads us to our last field season, because we're also investigating other sites. We'd like to have different data points where we can look at elevation through time, and that is the site known as Nazareno. And this is a picture of Nazareno. Nazareno is also in southern Bolivia. It's only about 60 kilometers northwest of Quebrada Honda. It's not very far at all as the condor flies. Also, it's in the eastern cordillera of the Andes. The elevation is similar. It's about 3,600 meters. Turns out Nazareno is a place that I had been aware of for a long, long time, but really hadn't had too many opportunities to visit. When I was at the university in Potosi, where my colleague Federico moved to after that conference in 1999, I was there in 2007 and looking through the collections, and I saw this specimen. This is from another plant-eating animal. And I noticed on the tag, it said Nazareno, and this had been collected back in 2003. He had been there with some researchers from Duke, and they had made a small collection of fossils. And some of them were very, very interesting, particularly a rodent that ended up describing some 15 years later a new species of chinchillid. But those folks weren't the first people to find fossils at Nazareno. It was this guy named Castellanos. At least he was the first to describe the fossils. There was a train station at this town of Atocha, located not too far from Nazareno. And this skull, which is from an armadillo, was found not far from the train station, and he described it. And if you translate the title, it's basically a new extinct armadillo from southern Bolivia, and he named it Dasypodon atavus, new genus and species. And this probably is from the same outcrops as Nazareno. We don't know for sure. We don't have great provenance data for it, but based on the maps, we think this probably is also from this site. That's really the first discovery. But then no one did anything with this site until this guy named Yoshinori Owiso published a paper in 1991. And he's an interesting guy. He was basically on the Japanese version of the Peace Corps there in the late 80s and didn't have a whole lot of geological experience, but he did some basic geology there, and he collected some really interesting fossils and then published this a couple years after that in an edited volume. And these are examples of the fossils that he found. Some nice specimens, some skulls there on the upper left of plant eaters, some rodent jaws down in the lower left. On the right there, some of these armored mammals, different types of armadillos, including horned armadillos. Unfortunately, these were subsequently lost, which can happen. He was working not with the Natural History Museum, but with the Geological Survey. And who knows what happens to these fossils. I followed up with him by email much later, followed up with other folks, and unfortunately, these are all gone. So all we can do is speculate. But we were able to go there in 2008, again with Carmi Garzione, to do a little bit of prospecting. We did some basic looking at the geology. They took some samples. Interestingly, they must not have panned out for the clumped isotopes because that's never been published. But we did find some decent fossils there. This is an example of some small lid upturned fossils. Oops, went too fast there. There we go, circled. There are teeth there in the lower right. In the upper right there, we have an ankle bone. In the lower left, we also have some limb bones. And these are relatively, this is from a small lid upturn, small dog-sized animal. These are glyptodont shell bone pieces, much more fragmentary than what we saw at Quebrada Honda. We did get to go back briefly in 2017 to try to do a little bit more geological study and collect a few more specimens. We weren't able to spend a whole lot of time there, but we did find some interesting things, some nice meat-eating mammals, some armadillo shell fragments, and this beautiful little jaw. This is a tiny jaw. You can see my hand there for scale. This is another plant eater, but really a very small, one of the smallest ones around at this time. It's a Intratherium is the group, another one of these endemic plant-eating mammals. We found the front part of the jaw there too. That's what the whole thing looks like. And this turned out to be another new species that Federico and I named just a couple of years ago, Gechuiciu arenalaensis. And Gechuiciu means little hoof, in allusion to the fact that these were very, very small ungulates, and arenalaensis just refers to this town of Arenales, which is the one that's close by to the site. And here's a little reconstruction of what this thing looked like, we think, with a dragonfly there for scale, and one of the larger notoungulates, although not a large notoungulate by any stretch of the imagination, there in the background. We have not yet recovered as many species of mammals from Nazareno. Part of this is due to the fact that we haven't done as much collecting there. We haven't spent as much time there. The more specimens to collect, the greater the chances are that you're going to find specimens of things that are rare. And one of our motivations for wanting to go back and spend more time at this site is that we've got a pretty good diversity of species now. And certainly if we go back there, we're going to start to get closer and closer to the number of species that we have at Quebrada Honda. With that in mind, we went back in 2022. You'll see Beverly right there in the center. Federico's there at the left, along with a student at his university just to his left. I'm obviously there on the right, and in between Beverly and I are two students. We had the opportunity to take two geology students with us to collect some samples, help with some stratigraphy. And just next to Beverly on the other side is Naomi Levin, who is up the road at Michigan. And she is an isotope expert. And we're hoping to incorporate her expertise on some studies in Nazareno and other places. And we were able to get to spend some time there, mostly focusing on the geology, not so much the fossils, but it is impossible to go to a place that has fossils and actually not collect fossils. You just can't avoid your eyes. We did find some things that turned out to be pretty interesting. One of the things we really need to do at Nazareno, which we have done well at Quebrada Honda, is figure out how old are the fossils. We have a general idea, but not nearly as good an idea as what we would like. The Nazareno Formation is the upper part of this diagram, and it sits on top of the Tupiza Formation. So these blue bones represent where we find fossils, basically throughout the Nazareno Formation. In the top part there is an ash, which we have not found and dated. We think we know what ash it is, but this was published by other authors, that has a date of about 12.8 million years or so. That tells us that the fossil bones are older than 12.8 million years, but they could be, 12.8, 12.9, putting them as the same age as Quebrada Honda, but they could also be significantly older than that, because the only lower constraint that we have is from the Tupiza Formation. This is a more volcanic formation that underlies the Nazareno Formation, and we have some dates from there that are basically 21 million years old. But that's a pretty big time range. We're talking a 7 million year interval. And for fossil mammals, that is pretty imprecise. Here's another way to look at it. Here's a picture of the Nazareno outcrops. We find fossil bones in all of these areas here. Down below, we have the Chapeza Formation, which is around that 21 million. This is the range if you include those error bars on there. And then this ash, is way up here in the section somewhere. What we'd like to do is find other ashes in between so we can narrow down how old those bones are. And that's one of the things that we did when we were there. We looked for things like this. This is what a volcanic ash or a tuff looks like. This is important for radiometric dating because if you're familiar with radiometric dating, it is based on the idea of radioactive decay. And essentially what happens is these radioactive materials are spewed out with volcanic eruptions. It can be with like a lava, like a basalt, or also with ash. They cover the landscape, but then those minerals start decaying. And you measure the proportion of parent material to the decayed material and use it to back calculate how long ago that happened. Volcanic rocks, tuffs, lavas, those reset the clock, so your parent material starts at 0. You can't radiometric date, typically, where you actually find the fossils in these soft rocks down here because you don't have that resetting of the clock. You don't know what parent material you started with. So you go through and look for things like this, volcanic ash, and take samples. Hopefully they're good quality that you can then do radiometric dating on. And the students and Beverly and Naomi, I helped out a little bit here and there. We did lots of looking for ashes and then measuring the geologic sections, because if you're going to take samples, you have to know exactly where they are within the rock layers. This is not only true for fossils, but also for isotopes and really anything else. And no one had ever done this since Owiso and back in 1991. And his sections are good, but they're fairly coarse. And that was a long time ago. And we don't know exactly where they correlate to different sites that we visited. And we spent a lot of time doing that. It's climbing over rocks. It's describing the rocks. It's digging them out so you can see them. It's using this thing called a Jacob's staff, which is basically a fancy yardstick or meter stick, as the case may be, with this little sight on top that allows you to know when it's level, because you're going up these angles and you need to measure the exact thickness of these beds. And you work your way up and describe it, and then you can put your samples within that context. And the idea is that you can create something like this on the right, which is a stratigraphic column, a description of the thicknesses of rock layers, the types of rocks that you find at each layer, and the elevation where they are within the stratigraphic section, and where your ashes are located, et cetera, et cetera. The other thing that we took samples for was geomagnetic polarity dating, often just referred to as paleomagnetism. Paleomagnetism is a technique for measuring time that's based on the observation that the North Pole flips, or I should say the magnetic pole flips. I guess north is always north, but the magnetic pole is not always the North Pole. The Earth's magnetic field reverses every so often. Not exactly sure why this is. There are folks in the geology department that could certainly tell us more about it, but this is, why does this happen? Not exactly sure. We have some idea of how it happens, but it's totally, I think, unpredictable. It's random. It doesn't happen every, 10,000 years. It's basically random. And this is what our magnetic field looks like normally, but then when you undergo these reverses, everything goes haywire, and then you end up with the magnetic pole being in Antarctica rather than in the Antarctic. I think there's some movies about natural disasters happening when this reversal takes place. That is science fiction. I don't think we exactly know, but it's probably not as bad as in the movies, which is probably a good rule of thumb. And it's essentially random. It generally occurs every 10,000 to 100,000 years, but there is no pattern. And this turns out to be very, very useful, because this signature is recorded in certain types of rocks, and we can measure it and correlate it to our master scale to help us get more precision in age estimates. And let's walk through how that works. I think it's kind of interesting. This is the geomagnetic polarity time scale for the Neogene, which is the past, 26 million years or so. Present is up at the top, all the way down. Older is towards the bottom there. That column there, that polarity column, shows you when we have a normal polarity, which is one where a compass points towards the North Pole. That's in black. And it shows you where we have reverse polarity in white, which is when that same compass would point towards the south. And hopefully you agree, if you look at that pattern, there's no pattern. It's essentially random. People look at this in detail. They measure this. This is a chart version of what we just saw. I know it's small, and I'm not going to focus on a lot of this. What I want to point out is that each of these black and white intervals has a name, which is a letter code, essentially. We have a very precise beginning age for it. We have an end age for it, and we have a duration for it. And when I say precise, these are in millions of years. If you can read the beginning date, that's 5.235 million years. That means that we are measuring, in thousands of years. Essentially, 5,000 years is what that last digit is, which is pretty darn precise when you're talking about millions of years ago. How do we get those precise dates? We do that by looking at the master record, which is produced at the mid-oceanic ridges. We've come back here to a picture of the Earth, and this shows topographic relief on the ocean floor. And the red there is the mid-oceanic ridge that you see between North America and Europe. And if we zoom in on it, this is a diagram to show you that new material is constantly being made at these mid-oceanic ridges. It's where magma is coming out and being pushed out, and it's a constant record. And that's nice because it'll record when we have normal polarity, and then when we have reverse polarity, and then when we have normal polarity, etc. And scientists who look at this can give us a very complete master scale of what happened on the Earth over the past, certainly the Cenozoic, and it goes back even farther than that. In paleontological sites like Bolivia, we just have a snapshot. Fossil accumulation, rock accumulation on the continents is sporadic. We just get these snapshots essentially compared to the middle of the ocean. But we can take that snapshot and try to correlate it. Here's an outcrop. We can imagine putting together a geologic section, finding bones throughout that section. And then we go through and we take paleomag samples. We take samples of the sediment, and if they're collected well, and if the sediments are appropriate, it will record which way a compass would have pointed at that point in time, towards the Arctic or towards the Antarctic. And we can measure this and we can code it as normal or reverse. And we can imagine us going through and taking samples every 5 meters or 10 meters, analyzing them and categorizing them as normal or reverse. We do that through our section, and we can end up with a little barcode that looks like this, of reversed and normal intervals. We can come up with a hypothetical local section like this. You can again see there's no clear pattern, but we do have black and white, normal and reversed intervals. If we have a radiometric date in one of these intervals, let's say here we've got about 12.8 million within this basal reversed interval, that's all we need to tie this to the master pattern. Here's our global scale. We can correlate these patterns like this. And now if we have a bone here in our third reversed interval, we can't just, we don't just say it's younger than 12.8 million. We can correlate it to the specific chron and say, it turns out it's between 12.5 and 12.75 million years, which is a lot more useful. Therefore, even if you can't get lots of radiometric dates, this is another way to get the precision as if you had lots of radiometric dates. Therefore, we took, and we meaning Naomi, took paleomag samples at Nazareno. And this is the way that you do it. Because we're looking at directions and orientations, you have to be very careful when you take these blocks out. You carve out the rock, all the eroded rocks, so you're getting to a fresh rock face, And right there, she's using a file essentially to smooth out that surface. She uses a special, essentially geologist's level and compass to measure the exact position and direction of that hand sample that she's taking in the rock outcrop. And she'll mark that on there, mark which direction that face is going, what its angle is. So when it's back in the lab, the person who's analyzing it, who in this case was Dan Pepe at Baylor, another of our colleagues, knows exactly how that was positioned in the rock face, even though now it's totally removed from that. You then carve that out, make sure those are well indicated on there. You wrap them up, tape them up, and then you bring them back where they're processed in a fancy lab that has to be magnetically isolated, right? Because we have magnetism from all sorts of things, and you have to have special facilities where you can analyze this and not have it altered by other magnetic fields. And our hope is that we'll start to get some paleomagnetic profiles for Nazareno to help us narrow down the age. Of course, we did look for some fossils. We found some fossils. This is one site that we found on, let me think about this. It was the second to the last day. It was the last day for a chunk of our team who had to head back earlier. And I found this fossil, which didn't look like a whole lot, didn't have teeth. So usually I'm not so interested in things that don't have teeth, but they can be interesting. And I took a closer look, and this turned out to be part of a turtle shell. We started following it up slope, we excavated it, and there was more there. This is Federico excavating out. And this is him and his student collecting. This is a plaster jacket there in white, what turned out to be part of the shell of a tortoise. Now, this is not nearly as big a tortoise as the one we found at Quebrada Honda, probably closer to a red-legged tortoise, which is what type that lives today in the tropics of Central and South America. But this still is not the sort of animal that would probably be living at elevation. And hopefully we can find more animals like this, more ectothermic animals that can give us some independent information about the habitats there at Nazareno. This is the first time when we'd gotten animals like this. We also found a microsite, which is really useful. This is a handful of fossils, literally speaking. Many are tiny. You can see I have a some of them, I was going to say handful again. Several of them noted what they are. Really tiny rodent jaws. Those are from a thing probably called Acroechemes. Some rodent teeth from relatives of chinchillas. A sloth toe from a not very big sloth. A little marsupial tooth there. These are the first non-carnivorous marsupials that have been collected from the site. And we're hoping that going back to this site, we can maybe do what's known as screen washing, where you collect the sediment, you wash it out, and you get lots more of these teeth, because they can give you lots of insights into tiny animals that you're very unlikely to find when you're just walking around looking at the ground, which is what we typically do when we collect vertebrate fossils looking for bigger things. Hopefully, this will be a really nice insight into the sorts of little animals that were living there. It is a much bigger basin than Quebrada Honda. There are lots of possibilities. This site where we found the tortoise and these microfossils was really quite far in terms of kilometers from where Oisa worked and where we went in 2007 or 2008 and 2017. So there are lots and lots of possibilities. And the idea, the hope is that we'll be able to head back there next year. We're trying to get some financing to allow us to do that, and also have more student projects. Evan, a PhD student here, has his background and master's degree in paleosoils. And clearly that's something that we'd also like to do at this site and potentially some other sites. If you want to learn more, I have a blog. I have been bad about adding new entries since the pandemic. But before the pandemic, I think there's some pretty good ones. And at some point, I'll get back to it. But you can check that out, raftingmonkey.com. That alludes to the title of a book. I put out a popular science book, Horned Armadillos and Rafting Monkeys, back in 2016. And if you look on the cover there, you see illustrated by Velizar Simeonovsky. That was the impetus for doing a lot of these illustrations. And it's nice that I can use these for talks too. You can check out my homepage. I have been tweeting, given recent changes in Twitter. I haven't done that lately. I'm not sure if I'll continue or not, but anyway. And last but not least, if you want something that's a little bit more technical, I will highlight a volume that I co-edited with Scott Simpson, who's on here, and Denise Sue, who I suspect all of you know, who was curator at Cleveland Museum of Natural History. She's a paleoanthropologist and paleoecologist. She's now down in Arizona. We had a conference we hosted at the Natural History Museum, and we put together this textbook, kind of textbook. It's an edited volume, Methods in Paleoecology, and this has chapters that talk about many of the techniques that I've highlighted here today. We have an excellent one on paleosoils. There are some on phytoliths, on all sorts of stuff. It is one of these digital books. You can get printed copies if you want, but especially for those of you who are academics and can access Springer through Case Western Reserve University or through another institution, you can go there and you can download a PDF of the entire book. So if you can't do that, let me know and I can set you up with a whole book or other chapters. But that's been a great resource. We hope it's a great resource for students. But that, as I said, gives lots of details on the things that I mentioned here today. It's a little more technical, certainly, than what you get in horned armadillos and rafting monkeys. I do need to acknowledge funding. We can't do any of this without money. Case Western has been a great supporter. They're the ones that funded our trip in 2022, the College of Arts and Sciences, when Expanding Horizons grant. National Science Foundation has been a long-term supporter of us, and we'll be asking them for more money again, as has National Geographic. So thank you to all those funders. And I do also want to say tons of collaborators that I'm not calling out, I haven't called out specifically in this, but I don't think any of these species that have been described in isolation. Often students, colleagues, et cetera, have been involved in this. So lots of people to thank, and also with the fieldwork and logistics. That is all I have. So I would be happy to open it up for questions, comments, criticisms.
Great program, Darin. Thank you very much.
Wow.
Thank you. I enjoyed it. I'm going to see if I can, my controls go so I can stop sharing and enlarge so we can see all of us. I do that.
So when you come across one of these little micro caches, you know, when you had all the little tiny bones in your hands, and do you have like any kind of hypothesis or explanation as what created that? Was it just a little local fissure where a bunch of animals died, or was it a result of erosion and a you know, recent erosion and deposition, or what do you think? What do you think results in that?
Yeah, in the case of this one, and a lot of them that we find, we think it's probably the latter, and that it's things that accumulated throughout time in these ancient soils and these other things, and that you've got the topography that allows the smaller sediments to be washed away, but that don't allow the small bones and teeth to be washed away, and that they're accumulating there. So it's like a natural sieve, essentially, that concentrates these things.
Are the fossilized bones, are they of similar density to the material that they're encased in, or are they a little lighter? Are, you know, is there any difference?
It is a good question in terms of absolute density. I will say the bones are permineralized to some extent, so they're denser than regular bone. The teeth are essentially the same, chemically unaltered, which is what makes them so nice for these isotopic studies. Mm-hmm. The rocks are not really heavy, you know, super dense rocks, mm-hmm. and they're usually somewhat unconsolidated, and they'll be eroding out of these. Mm-hmm. So I think they're probably close in overall density to it, but once you start to erode the rocks, the sediments themselves are much, much smaller, and so they individually can be washed away. Whereas if you're in, you know, rocks that are pressed together more, you don't have that effect. Like in Chile, we've got really hard, really dense rocks. Yeah, we don't find any microsites like that because the bones are in the rock, and you collect them that way, and you have to physically take the rock off. But a lot of these specimens are what I call auto-prepped, where nature does the erosion, and you'll find it, and it looks like it just died there yesterday because the sediments can loosen up and wash away.
So the great geologist in the cosmos is working for you on that one.
Exactly, yes. Yes, sometimes they'll create, sometimes they destroy, but yeah, it's best when they do the work for us.
Are you getting back down there every year or is this sort of hit and miss as funding and your own schedule allows?
We'd like to get there every year, but it definitely depends on funding. When we get big grants like National Science Foundation grants, those are typically 3-year grants. And then if we have a reason, if we have funds, it's usually pretty easy to extend that for a 4th year, sometimes to a 5th year even. So when we have that, we'll go every year for several years. Sometimes there'll be a gap in between. With smaller grants, they're often just enough to fund 1 year. And the idea typically with those, like with this EHS from Arts and Sciences, the idea is that we can collect some data that we can then use to get a bigger NSF grant. So the hope would be, we had a bunch of years for Gibraltar Honda where we went more than half a dozen times with a couple grants, and that's kind of finished. And now we're in the preliminary data collection stage for Nazareno, and the idea is that we'd get an NSF grant. And if we were able to do that, we would go for 3 or 4 years in a row.
Your South American partners, do they go back to these sites in the years that you can't go there? And do you have a regular presence at the sites?
We do not. No, the way that we work, and it varies a lot in different places, South America, different sites. These sites, Bolivia, there aren't many paleontologists. There's basically Federico. There's some folks in La Paz right now. And when he's not going to the field, he's teaching classes. He's in the School of Geological Engineering. And usually it's our funding that will pay for being able to get out to the field and things like that. This is about a 5 to 6 hour drive from Potosi. So it's not a huge, drive, but you have to go there, you have to camp. So it's a little bit of an expense that they don't typically have the money for. Sometimes when we're down there, though, if we can spring for the field vehicle and other things like that, the university will be able to contribute a truck and a driver to help, especially if we want to bring some students and things like that.
So do you ever get raided to these sites ever? I think about, dinosaur sites, and given the very real value the dinosaur bones have now, and lots of other fossils, there's tremendous interest in this commercially. Do you worry about that, or is the nature of the stuff you're going after, it's not necessarily the most spectacular stuff, even though scientifically it's a gold mine?
I say in the back of my mind, I worry about it. In Bolivia, it doesn't, at these sites, it does not seem to be so much of an issue, partly because they're pretty isolated and we don't tend to find a lot of really photogenic fossils at these sites. At other sites, there's a really well-known one among paleontologists called Tarija, which is right near the town of Tarija, which is also in southern Bolivia, a big town. It's the department capital, but they're Pleistocene. It's a famous Pleistocene site. And these mammals then, are in the tens of thousands of years old. And that's where you get giant ground sloths, you get giant glyptodonts, you get saber-toothed cats and things like that. And it's right where the town is. And that is a big threat. You get lots of collectors that go there specifically to get those fossils. They're exporting them illegally. I will say Bolivia is like a lot of countries in South America. I think the US is really an outlier in this regard, where the country owns the vertebrate fossils. Doesn't matter if you own the land, that is national patrimony. Here in the US, you own the land, you own the fossils, but that's not true down there. So anyone that goes there and collects and does anything with them, that's against their national laws. But still, people do it because, as you say, there's demand, particularly for these big photogenic macrofauna, and it can be a good way to earn extra money where it's tough.
Who owns the property where you're collecting? Is it like national, like BLM land in the United States, or is there comparable?
That is a very good question. I think I don't know how it's all set out. I'll tell you how we work is we go and our permit comes through the university. So it's a very diffuse, that Bolivia now is the plurinational state of Bolivia is technically the name of their country, and recognizing that it's really an agglomeration of different nations. And there's a lot of autonomy in the departments as well as in the towns and populations there. So we'll go to the town, talk to the mayor, and make sure that they are okay with us working there under the assumption that they really see the lands in that area. And I don't know exactly how it's worked out, but that's what we do. We cover basically everyone who is in contact with the folks who work there to make sure that it's okay. And then with individual landowners, same thing. We'll go and explain to them what we're doing and see if they're okay with it and do it that way.
I mean, two things that come to mind, and I'll finish this land ownership thing first. And haven't had a background in buying land in the United States and trying to protect land and whatnot. You know, I've followed a lot of international efforts to protect land, and Patagonia and other countries, you know, Northern European countries too. And you know, I can't help but wonder, in some of these governments, the structure is such that you know, it's like a paper exercise. You know, buying this, you know, for an agency like the Nature Conservancy, I don't mean to disrespect for any efforts, but an agency like the Nature Conservancy coming in and buying, you know, a big tract of land, and they take the money and they put this easement on it, and you know, and at the bottom line, it's like the land is government property. You know what you've done is you've infused some cash into there, but have you really resulted in any kind of meaningful and long-lasting thing? So that's what prompted me to ask that question, because many, you know, Cuba, for example, has the highest percentage of protected land in any country in the world. And it was because Castro, if he pulled into an area and he thought it was beautiful, he just declared it a national asset and said, you know, it'll never be developed. You know, so I was fascinated with that whole notion of where you're doing this work, and I appreciate your response to that. The other thing that came to my mind, you know, when I was listening to you and your passion and your commitment for this, I just think it's so sad that so many conspiracy theorists out there, you know, the folks who think that the UN are out to, you know, take over the world and whatnot, have just never had exposure to a true scientist like yourself, who's driven by you know, the passion and acquisition of knowledge and understanding things. So, you know, I really do appreciate what you're doing, but the enthusiasm in which you're doing it. And it's wonderful that you're in a position where you can pass that on and encourage that to students and whatnot. So just keep doing great things.
I very much appreciate that. Yeah, I do love it. Hopefully that comes across. My students in my mammals class can certainly attest to that. I get a lot of excitement, especially for teaching I love teaching anatomy. Also, I don't want to take anything away from that, but you know, mammals are what I've always been interested in. And I will say, for me, the other thing I really love is the ability to keep learning. And you know, that's one thing, especially working with all these geologists like Beverly and other experts. It's been great for me because I'm always learning. It never gets dull.
That was just a brilliant thing. That little Venn diagram of geology and biology leading to paleontology. That was the most elegant explanation of that I have ever seen. So that was just really brilliant. I will be quiet here after making a pitch for our next program. Stay tuned for details coming out for that. And it's going to be on longhorn cattle in Ohio. And anybody else have any other questions and whatnot, chime right in here. And if not, Darin, thanks so much for really kind of a mind-blowing, multi-dimensional. And I'm still getting over the thing that you're in the medical program. Holy smoke. All right, but fantastic presentation. Thank you all for joining us. And thanks again.
Hello?
Yeah.
Darin, I just want to thank you for an excellent presentation. That was so good. I also have a question regarding the magnetic changes. Is there any indication of possible impact of climate or weather in those different changes in the magnetic field?
So with the caveat that I'm not up on everything, I think not. I think the idea is that these are generated deep within the Earth's core, and that it's coming out from there, and that there are changes that are taking place there that then result in the changes in the field, and that anything in the weather related that's just right on the crust would be too superficial to affect it.
Okay, thanks.
Yep. And thank you for your commentary on the presentation. I appreciate that. Great. Well, thank you very much. If no one has anything else, I really appreciate the invitation. I love doing things like this. As I said, I love seeing all these familiar faces, and also nice to meet those of you who I've never met before. Hopefully I will see you in the flesh at one of these talks or elsewhere.
Okay, thanks so
So
much.
long all.
Okay, bye-bye.
Bye.
Bye all.